All right, let's get started for the next section. Out of all the abstracts that were submitted for this meeting, 3 were identified as outstanding abstracts, and those authors were asked to give oral presentations. Our first presentation is Elizabeth Wong, who is a 4th-year graduate student in immunology with Dr. Shijing Li. She is studying the etiology of an unusual subset of CD8-positive cytotoxic T cells that are class II restricted. And the title of her talk is Conversion of Effector CD4-positive T cells to an MHC class II restricted CD8-positive T cell lineage. Elizabeth?
Thank you. Good morning. Thank you for the opportunity to present today. Over the next few minutes, I would like to share with you my project on the conversion of effector CD4 T cells to a novel CD8-positive MHC class II restricted lineage. CD8 T cell mediated immunity is emerging as a promising focus for HIV vaccine development. Recent studies on rhesus macaques have demonstrated that CMV vectors expressing SIV epitopes can elicit CD8 T cells that provide potent and long-lasting protection against SIV. Intriguingly, these protective responses were dominated by CD8 T cells that recognize diverse, unconventional SIV epitopes in an MHC class II restricted manner. The origin and ontogeny of this non-canonical population remain elusive and will be crucial insights for the development of new HIV vaccine strategies. Today, I will not be able to tell you the origin of MHC2-restricted CD8 T cells generated during SIV-CMV vaccination, but I would like to tell you a story from our mirroring studies that may provide important and exciting clues. We serendipitously entered this field of work when we discovered a population of murine effector CD4 T cells with a lineage phenotype mirroring the one generated during experimental SIV-CMV vaccination. During our study of microRNA trafficking during CD4 T cell activation, we observed that murine CD4 T cells deficient in the vesicle trapping regulator, VPS34, dramatically lost CD4 expression upon in vitro stimulation. Importantly, we could not merely attribute CD4 surface downregulation to defective surface-oriented trafficking, since CD4 did not accumulate intracellularly. Furthermore, a proportion of these VPS34 deficient cells began to express surface CD8 alpha beta. While multiple lineage intermediate effector CD4 T cell populations have been characterized by other research groups, this CD4 negative, CD8 alpha beta positive, XCD4 positive T cell phenotype has not been previously reported. Our XCD4 T cells also began to produce granzyme B and interferon gamma, which are hallmark CD8 T cell associated cytotoxic mediators. Importantly, we observed that wild-type effector CD4 T cells used for experimental controls could also give rise to XCD4 T cells that produced cytotoxic molecules. This indicated to us that this population was not a knockout-specific phenomenon, but instead reflected fundamental reprogramming of lineage specification induced by CD4 T cell activation. Taking our findings together, we hypothesized that effector CD4 T cells could undergo lineage conversion to an XCD4 T cell population characterized by CD8 T cell-like identity and functional capacity, as well as MHC class II mediated recognition. We noted that demonstration of true lineage conversion was crucial to validating our hypothesis. To accomplish this, we took a genetically based approach. Our group had previously developed a mouse strain that expresses a Cre recombinase transgene under the control of the OX40 promoter, as well as a transgene for the fluorescent reporter TD tomato under the control of the floxed Rosa stop cassette. By this design, TD tomato expression is induced only when the co-stimulatory molecule OX40 is expressed. Because OX40 expression is preferential in activated CD4 T cells, this system would allow us to demonstrate that XCD4 T cells originate from effector CD4 positive T cells, and that they do not originate from a pre-existing trace population of unconventional CD8 T cells. Upon in vitro stimulation, we observed that FACS-purified CD4 T cells expressed TD tomato in a dose-dependent manner, indicating that TD tomato is indeed a faithful and sensitive marker of activated CD4 T cells. Among TD tomato positive effector CD4 T cells, we found that a small proportion of cells highly expressing the activation marker CD44 downregulated CD4 expression, and that the majority of this population expressed CD8 alpha beta. Interestingly, we noted that some CD44 high, CD4 positive effector cells also express CD8 alpha beta. Considering the other lineage phenotypes previously reported, we began to consider that our xCD4 T cells might belong to a collection of lineage intermediate phenotypes derived from effector CD4 T cells. Collectively, we interpreted this evidence as convincing support for a CD4 negative, CD8 alpha beta positive T cell population that derives from lineage converted effector CD4 T cells and not from aberrant CD8 T cells. We then sought to determine whether lineage conversion to xCD4 T cells could be induced in vivo. We began by inducing a robust antigen-specific T cell response against acute Listeria monocytogenes infection and wild-type and VPS34 flocks CD4 Cree mice. These mice express the LLO118 T cell receptor transgene, which recognizes the major Listerio-lysine epitope of Listeria, presented in the context of MHC class 2. We found that Listeria-infected LLO118 VPS34 CD4 Cree mice generated a substantial population of CD44 high, CD4 negative effector T cells, similar to what we had observed in vitro. Also, as we had seen previously, a proportion of the xCD4 T cells express CD8 alpha beta. We also found other lineage intermediate phenotypes, namely CD4 CD8 double negative, CD4 negative CD8 positive, and CD4 CD8 double positive, providing further support for the idea that effector CD4 T cells can give rise to a spectrum of lineage phenotypes. Furthermore, we observed that our CD4 negative, CD8 alpha beta positive xCD4 T cells could also be generated in Listeria-infected LLO118 wild-type mice. And in fact, these wild-type cells express CD8 alpha beta at a higher frequency than VPS34 deficient cells. This evidence confirmed to us that effector CD4 T cells possess a fundamental lineage conversion capacity that is enhanced in the absence of functional VPS34. We followed up on these results by asking whether xCD4 T cells could still be generated from wild-type effector CD4 positive T cells if the T cell receptor repertoire was unfixed. Indeed, we found that non-TCR transgenic wild-type mice acutely infected with lymphocytic choriomeningitis virus generated LCMV-specific MHCII-restricted CD4 negative T cells, even at a higher frequency than LCMV-specific CD4 positive T cells. Furthermore, CD8 alpha beta expression was induced in a proportion of these CD4 negative cells, but not in the effector CD4 positive cells. Accordingly, we concluded that an xCD4 T cell response can be induced in the physiologically relevant context of CD4 positive T cells with an open TCR repertoire. However, neither the Listeria nor the LCMV infection model allowed us to definitively demonstrate that effector CD4 positive T cells can lineage convert to xCD4 positive T cells in vivo. To achieve this, we acutely infected our OX40 Cree TD tomato reporter mice with LCMV Armstrong and analyzed the LCMV-specific MHCII-restricted T cell response. We found that both LCMV-specific effector CD4 negative and CD4 positive T cells were generated, and that TD tomato expression was induced in both of these populations. This result indicated to us that an xCD4 positive T cell response to acute infection does indeed derive from lineage-converted effector CD4 positive T cells. Given that T cell lineage identity is dictated by lineage-specific transcriptional programs, we then analyzed both wild-type and VPS34 deficient xCD4 positive T cells for the expression of a panel of CD4 and CD8 T cell associated transcripts. We found that the phenotype we had observed at the protein level was mirrored by transcriptional expression patterns. CD4 transcription was profoundly silenced, concomitant with a modest upregulation of CD8 alpha beta transcription. Additionally, transcripts of cytotoxic mediators were markedly induced. Still more importantly, we observed a reversal in the relative expression of master transcription factors THPOC and RUNX3. CD4 T cells characteristically exhibit THPOC high, RUNX3 low expression, while CD8 T cells exhibit THPOC low, RUNX3 high expression. We found that CD4 T cells exhibited a THPOC low, RUNX3 high intermediate profile, which indicated to us that this population has indeed shifted towards the CD8 lineage program. Given this transcriptional evidence, we concluded that the CD8 T cell-like phenotype and functional capacity of CD4 T cells is underpinned by overarching changes in the transcriptional programs that determine T cell lineage identity. We believe that our findings fill key knowledge gaps in both T cell and HIV immunology, and more importantly, that they offer a novel strategy for HIV vaccine development. We have defined a previously unknown CD8 T cell-like population that is generated by lineage conversion of effector CD4 positive T cells. We highlight that this population represents full conversion from the CD4 to the CD8 positive T cell lineage phenotype, and as such is the CD8 lineage extreme of a spectrum of effector CD4 T cell derived lineages. Having discovered this CD4 T cell population and demonstrated its ontogeny, the next important question is, what is the physiological relevance of these unconventional T cells? And for what purpose could we harness their generation? We cannot explain the role of CD4 T cells in all infection contexts, but we propose that CD4 T cells are uniquely suited to combat HIV infection. Specifically, their lack of CD4 expression would provide resistance to viral entry, and their MHCII recognition capacity would facilitate destruction of antigen-presenting cells harboring HIV reservoirs. We speculate that our CD4 T cell population may parallel the MHCII-restricted CD8 positive T cell population generated by CMV/SIV vaccination, and we suggest that these cells are also of CD4 T cell origin. Accordingly, we propose that an HIV-CMV vaccine designed to elicit CD4 positive T cell mediated immunity could provide potent and long-lasting protection against HIV. The next crucial step is to define the molecular mechanisms that control lineage conversion to CD4 positive T cells. We are currently investigating the transcriptional and epigenetic regulators of CD4 T cell lineage conversion, and whether these mechanisms also operate in human CD4 T cells during HIV infection. Our eventual purpose is to be able to offer a novel HIV vaccine adjuvant that can harness effector CD4 T cell lineage conversion and induce a robust population of CD4 T cells that can provide potent protection against HIV. I'd like to acknowledge the individuals, facilities, and funding agencies that made this work possible. In particular, I would like to thank my thesis mentor, Chi-Jing Lee, and our collaborator, Dr. Georgia Tamaris, for their guidance and support, and CFAR for the small grant that currently funds this project. Thank you for your attention.
Elizabeth, other than CD44, are there any other phenotypic markers that stand out among these CD4 cells, like they express CD40 ligand, PD1, any conventional markers like that? Have you looked at that? We
have not looked at those markers yet. We have looked at cytokine expression, so granzyme B, interferon gamma, and we saw the same thing that we saw in vitro. Those CD4 negative cells upregulate expression of interferon gamma and granzyme B.
Any other questions?
So in your in vivo models, have you tried killing assays?
No, we have not yet. We're working on an in vitro assay right now.
I have a question.
Yes.
So is this, are CD8 positive class 2 restricted cells a hallmark of CMV infection?
I don't think so. No. So actually, the way this population was discovered was with the Picker paper. By chance, that CMV strain had several mutations that promoted the production of CD8 T cell response that recognized unconventional epitopes in MHCII-restricted manner. So, as a general rule, I think this is a pretty trace population that does not really have a major role in combating infection.
Any other questions? No, thank you very much, Elizabeth.
Thank you.